Well, welcome everybody to today's JMP Academic Live webinar, Genomics Research with JMP Pro 17. I'm Ross Matusalem from the JMP Academic team. I'm just here to host and manage logistics today. Our speaker is Chris Kirchberg, a data scientist in life sciences here at JMP, a biochemist by training, and he's here to talk to us about using JMP Pro 17 to analyze genomics data. And this is a good topic with JMP Pro 17 having just come out this past October 2022, and JMP Pro kind of changing the way that genomics analysis is done using JMP tools, whereas before it was in the separate JMP Genomics platform. We now are introducing tools and enhancement to existing platforms in JMP Pro to handle genomics data. So before we hand it over to Chris to get started, I do just want to make a couple announcements. For those of you who've engaged with us before, this will probably be old news, but for those of you who are new, we want to let you know that the JMP Academic team is here to help you learn JMP and get what you need out of it. So if you're ever using JMP and wondering, how do I do X, or what tool is even the right tool to tackle the problem that I have, we're here to help you. And that can be in the form of just a quick one-on-one support call, or maybe even having a workshop or training session for you, your colleagues, students, research assistants. If you want to arrange any kind of support whatsoever, please just contact academic@jmp.com to get started. And also, for those of you who might teach with JMP or are thinking about teaching with JMP, we recognize that when you're teaching in general, and certainly incorporating software into your course, that a lot of consideration goes into the materials that you use and the examples that align with, in this case, JMP. So we offer a range of free teaching materials at jmp.com/teach. So these are things like sample data sets, case studies with exercises, in-class activities, also a library of quick video tutorials to help your students quickly learn how to use JMP, so you don't have to spend class time on it. So head on over to jmp.com/teach if you want to find some materials for your course. So we're about to get started. As we do, I want to direct you in the Zoom interface to the Q&A panel. So you can see the little icon for that particular tool right there with the overlapping speech bubbles. Feel free to ask any questions as we go along in that Q&A interface. I'll be in there answering them as we go to the best of my ability, and then we'll probably kick some questions out to Chris at a couple points throughout as well. But please do use that Q&A instead of the chat. It just on our end helps us actually keep track of the questions in which have and haven't been answered so far. So with that, I'm going to hand it over to Chris. So Chris, welcome, and thanks for giving us a little time today.
Thanks very much, Ross. I'm going to go here and share my screen. And we should be able to see my desktop now. Ooh, yeah, let's get rid of some controls that are getting in our way here. There we go. All right. So thanks, everyone. Cleaning up some controls. So as Ross had mentioned today, we're going to be talking about kind of omic data analysis in JMP Pro 17. And as I'd mentioned, we're starting to move a lot of things into JMP Pro. And this is kind of the description that was given online for you. But really, my highlight here is we want to do something that's a little bit, more native, a single package instead of two things kind of bridged together. And more importantly, we can do this on Mac and on Windows now. Before, all of our, most of the more useful omic data analysis tools were just in a Windows only type access. So now we've kind of started bridging that gap, and you'll see further development continue in the JMP Pro for all this omic data analysis. Now, there's a lot that could be done. So I am really only going to cover the differential gene expression in GWAS more as an introduction for those of you I see that consider yourself advanced omic data analysis users. We won't probably cover some of the more advanced stuff you might find in JMP Pro. Those are more like the predictive modeling, or that might be kind of genomic selection type tools, but they're there, right? A lot of this stuff has kind of already been there, just been enhanced to allow a much wider data set, right? More variables than observations. Also, things have been sped up in that regard. And so keep looking out for this stuff. We're adding things right now. It almost seems like every dot release right now to kind of fill in gaps and continues on for further versions as we add more and more things to be more fully functional. So that's why I want to give you just set expectations for today. So I wanted to start with the differential gene expression. And I've got an example here. And this particular example, I'm going to kind of go by like a little workflow. I mean, this might be a little simplified, but I want to give you an idea of some of the things that we can do as we think about it. So I'll put things in blue as kind of more bioinformatics processes or bioinformatics tools. Remember, JMP Pro is more statistical analysis and visualization tool, although bioinformatics does use statistics, and this is a little bit more focused on the statistics part of things. So we're assuming a lot that you've already collected your data, you've already aligned it, right? You've already made in this case, you've already made your count data, or you've already maybe you've done the extracted expression values, and you're ready. And we're going to assume that you've kind of normalized it, transformed it already, or centered and scaled, kind of depending on where that data is coming from, what you'll do. So you're going to start there. And you've probably already done at this point some sort of read quality control. So you've already kind of filtered, through read depth. You've already looked at, the number of transcripts and mapped to the transcript level of things. So that's what you're looking at, or what have you chosen for your gene model? All that's been done up to this point. So we're getting something that counts, some sort of a gene identifier. You've got your sample information, right? Well, sample characteristics, or experimental design information. You'll have that. And what we're going to do is this rest. So we'll do some basic sample quality control, things that you might do, principal component analysis and clustering. You might go then into some modeling, right? And we'll call that differential gene expression, right? That's what we're going to go to in this case. So we might be using ordinary least squares or multiple regression. It could be logistic regression, right? It could be a couple of different things, depending on what it is you're doing. So we're going to be doing some modeling and then doing what some people call contrasts, or those comparator groups. So we'll be doing that. And then, of course, there'll be some filtering, some clustering, some additional visualizations. So all of that you could do in JMP Pro. And then usually your next step in that workflow is extracting those gene sets, and you go out for interpretation. Go out to some pathway analysis tools, some network generation tools. You can export all of that and continue on with that workflow. So we're this middle piece. That's where we sit, all right? And in this particular use case, we're going to look at cattle follicle development. And so that's what we're going to look at. And so this is count data. And so that's what we're starting from. So this is the case study. Again, I'm not going to read through all this stuff, but there's a little link for a journal article. I even have a link for the data, right? So that's what we're going to do. We're going to pretend we're going to follow along. So here's our data, right? We already have in this case, I've taken the data. I'll talk about that in a little bit. But just to give you an idea of this data set, it's looking at different tissues, right? Granulosa and theca, and then looking at different stages of follicle development. And we're looking at different kind of animals, whether they're considered heifer or cow. And so that's different metabolic states. We're trying to understand here a little bit of what might cause an increase of infant mortality or embryo mortality in this case, because it can really reduce birth rates in calves. And we want to understand that. Why is there a reduction? Is there some metabolic state that's kind of reducing this? And if we can figure that out, maybe we can change diets. We can do whatever it is that we can to increase these birth rates, right? So understanding things at a molecular basis sometimes is helpful, especially in those tissues that would contribute to the birth of calves. So what do we have? So I've just taken a quick summary, right? And again, this is just built, by the way. I just use this to let you know how I did this. I just went to this tabulate. It was a pretty easy thing to build. And I just said, here's my tissue, right? I just wanted counts. I wanted my stage. I just kind of dragged and dropped it off to the side. And then I looked at my animal type. And there we go. So now I've got an idea of what my balance, if this is balanced or not. And I can quickly throw my aggregated statistics on there, and I'm done. So I just reproduced this little graph, this little tabular view I have behind me. And that's what we're looking at, right? And so again, it's just a good way to always verify what your data is, what you're looking at, how many you have, is it balanced or not? And that's what we've done. Very quickly done that. So we have 65 observations, but we're going to have, 28,000 some transcripts to evaluate. And that's a lot. All right. So that's what we have. And I'm not here to reproduce the article's results, just kind of give an example of how you might approach this. So first thing is data preparation. Now, I got the raw counts from Geo, and that says GSC identifier there. It could be normalized using DESeq2's median of ratios. And you could, do this in R, right? And then you could export it and log2, X+1 transform it, or the raw data, or you could create the R logs or VST transformations of these things. And it's kind of recommended here according to the DESeq2 documentation. So I've done all that, and I'm just going to use the R log exported values. And so that's what I did. So I already did that. And then I used the Geo series matrix file, and I opened that up. And that's what gave me all my kind of sample characteristics, right? So that's all these things right here, right? And along with that animal ID. And then I just kind of transposed and then merged them in, right? And that just kind of gives you an idea how I've gotten this, right? And so I just took two data sets, transposed it. So on my gene IDs were columns, my samples were rows, right? Here's my identifier. And just to point out, these are table manipulations that have existed in JMP for a long time, right? And that's all I did is just use this update when I had two tables. So I brought all that data in, and then I grouped it. And so for those of you who are not familiar with JMP, we do have this little tool that when you select a bunch of columns, I can right-click and say group. I group these columns out of convenience. I just called it mRNA. That's it. That's what our starting point is. I did do some extra manipulations for convenience. I made an ID column, just really easy to find each individual row with some nice, sample annotation. And all I did is just grab these columns. I clicked that one. I right-clicked on it, said new column formula, character with an underscore. All right, so nothing crazy, but just very quick, convenient ways to make columns or feature generation, as we'd call it. I did the same thing here because many times, instead of doing this as like a full factorial, expressing it, sometimes you can just concatenate two columns, did it the same way, and we'll just look at these as our comparators, right? Effectively the same thing, but turns it into a single level instead of a multi-level, and that works as well. That's just as functional, right? And so that's all the manipulation I've done to this point to get it ready. Did use a bit of JMP's tools already, but didn't want to go into great detail on how to do that. Just let you know what I did in JMP to get there, right? So that's simple enough. The real part is the QC. So the way I'm gonna go about this is I'm gonna go through a series of buttons to do things and then kind of point to you where they are, just to do my best to show you where I'm going, and then there's a little bit of how I got there. So first, I have the habit of marking and coloring rows. It's just easy for me to identify them later. In this case, we'll mark by tissue, so open or filled markers. The animal type will either be a triangle or a circular, and then the stage will either be colored by yellow, green, or blue, right? Again, very easy from a plots point of view to be able to track everything. So I've done that. I'll click on this little button, and you'll see that I have these little, what we call row states. Each row has a marker, right? It has a color, and as whether it's filled or open. And so it gives you an idea, like with heifer, how you can see they're different. And what I did for a lot of that, I usually went into the rows menu, and you could manually color and marker these things. It's up to you. I have a tendency to say row selection, select rows where. So I have a tendency to do that, and then color them based that way. Another way you could do that as a handy feature is just to use a color or mark by column. And so you could do it that way, right? And so that's a nice way to do that as well, right? And you have these options. So generally, this is kind of what I've done, just depending on what combination and order I've done them. and that's you get choice. So that's how I did that. So that's where I went to kind of mark columns and color by them to get all these markers. I just kind of saved that action into a single button. And if you want to know more, there's something called an action recorder or kind of an enhanced log. Feel free to reach out to Ross and Kevin on how to do that. And that's all I did. I just captured those steps. And then I just created a little button here. It's a little script that gets run. So I've done that. So next thing then is like, all right, let's see if there's any quality patterns, things I need to be aware of before I continue on. And sometimes clustering is a nice way to do that. So I'm going to do an initial hierarchical clustering. And here's what you're going to notice. We've enhanced JMP Pro recently with a much faster algorithm for clustering. Something you don't find in there. It's called a hybrid ward clustering, right? So we take advantage of k nearest neighbors for initial clustering. And once we've done that, we cluster within that. And you're not going to get much faster of an algorithm, even in the public domain through R, right? Because we just made it, right? It was just this in this past year. So this will be the fastest kind of hierarchical clustering method you can find. So it's called hybrid ward. And we immediately see a pattern, right? So although I've colored everything, this is where all the coloring, my columns is kind of helpful. I've added some additional annotation of tissue, stage, and animal type. Evaluate my clustering as well. But I noticed some pretty obvious patterns. There's a lot of missing data. This gray means missing data. So I probably need to filter out those transcripts that really have very little data or no data, right? They're basically zeros. So I need to get rid of that. Zero counts, right? In this case, zero R log values. And that's pretty much what we're going to do. There's other things too. It's proportion of missing we want to get rid of. So that's my first obvious thing to do. Right, so things that have nothing but missing data, and things that have a certain proportion of missing data. Right, and so that's what I can do. How did I get here? This is the Analyze menu. You look under Clustering, and here's our hierarchical clustering. That's how I've done that. And all I did is just say, I'm going to cluster. Right, I'm going to use all my transcript data. I'm going to cluster them. I'm going to be two-way clustering. If I wanted to do a missing value imputation, you could. We don't want to here. We want to standardize by our columns, and we want to use hybrid ward, right? So that's new. The rest missings are pretty publicly available references, but this is a hybrid version of that. And we have some help here for you if you want to learn more about those different methods right there. And that's all I did, right? Done. You could put a label. I tend to do that right here. That's why I created that text ID. And I use that as my label, right? And now I made it convenient to find my samples. So that was my first step, right? Got some issues. Great. Oops, here we go. So we've got that. It's good enough. So I want to start hiding and excluding columns. So I want to hide and exclude columns that have a lot of zeros. And I actually created a little script for this. So this is something that I'll show you here. When I click on it, wherever it went to, it didn't show up. There it goes. Let me cancel all of that. I clicked on the wrong button. Apologize. Done this. I want to hide and exclude. There we go. So I created this little script to say, here are all the columns I want to evaluate. So I add them. Right, here's all of them. And a portion of acceptable zeros in selected columns. So about 75%, right? I'm going to say is acceptable in zeros. The reason why is that some of these, right, looking at all these different categories, right? I have 3 levels by 2 levels. So there's 6 combinations that I could make. Looking at here, if I just was too strict, I could have 0. I could have only 10% of zeros. I could do that, and that might be nice, but it might be too restrictive. I want to be a little bit permissive, so I defaulted to 75% is permissible. I want to group them. I also want to group those that are too many, and let's hide and exclude those so they don't appear in any other platforms. So I created this script again. I can share this with you. It's not difficult, nothing proprietary here. And it's done. You can see it went through and now it made those. acceptable columns, right? Here's too many zeros, as I called it. So we filtered out about 5,500, and 28. I kind of hid them from view, right? So there we have it. That reduced a lot of them. Now let's take a look at it again. I could do that. Oh, it looks like I accidentally hid that. So I could cluster again on those things. And when I do that, I'll wait for a moment. It does this a little faster this time. There's no zeros. And we'll see that we don't have as much data there. But guess what? Even with those zeros there, it didn't have a huge impact on my clustering, which is good. All my theca cells cluster together. All my granulosa cells cluster together. I'll see differentiated generally groups together for both. Luteinized definitely will group together. It's pretty strongly different from the other two stages, both theca and granulosa cells. But when we get to differentiated and selected, there's going to be a little bit variability, but that's okay. And then within it, whether it's cow or heifer or not, well, that becomes a little bit more granular, but that's fine. They all look to be generally pretty good, and there's definitely some different gene expression patterns occurring here. So that's all good news, right? So first filtering done. We could do this by PCA if we want to. So there's just give you an end result, right? Here we are. And again, the reason why I color and mark things. The default, this is using a full SVD. That's something we've enhanced in JMP as well to make this much faster. So we can see this very clear clustering on our data here, right? You can see all of our granulosa up here, right? And you'll see down here, here's our theca cells down here, but are luteinized pretty clearly and pretty distinct, right? So far, so good. There's no real super large outliers. There's maybe out there. It's one way to look at this. And again, how did I get here? Again, we just multivariate methods under analyze. It's principal components. And again, pretty easy. Here's my WOG columns. I could use my text ID here as my supplementary. If you do nothing, it will prompt you. But we can be explicit and say wide data. And then we could do other things. Top 10, right? I could do all. We have special methods. We want robust or fast approximate. We can do missing value imputation, which we really don't have much of it. I don't think we have any missing data in this data set, which is great. And so we could do that. And that's basically what I did. The only difference here is I changed the default output. I only wanted to show scree plots and score plots because I think they're more meaningful. And when you have this large data set, that makes sense. Also, I'm going to go and save the columns. I can save just the principal component values. I did like the top 5, right? And I did right here. We saved out the top 5 of my principal components. Now, there's another way to do this. Some people will ask, oh, can you do this in 3D? We could do that. I used a 3D scatter plot. And this is nice since I saved my principal components. I could do it this way too. So I've got a nice view of my data set this way. And believe it or not, that's just going here to scatter plot 3D. And I could do it this way, but my Y columns, those are the principal components. And there I go, right? So sometimes I like to save out to do it that way. But you can also do principal components analysis within here. It's kind of your choice of where you start to do this, right? So both of them we do principal components. Sometimes you want to be more selective about which principal components are saved out or the method. So that's initial quality control. So far, pretty good. Nothing to worry about other than our, too many zeros. So we already excluded those. The other things we might want to think about is we might want to see the correlations between the two, right? I want to see the correlations of my samples to other samples. There should be a really good correlation sample behind this. So we need to kind of transpose this, right? Because everything is, I want to do this by that. I can't really correlate it to itself when I have genes as columns instead of samples as columns. That's just a simple under tables transpose. That's all that is. And so when I do that, I get a nice table like this, right? And that's all good, right? I've got everything here. And then we call it cluster and sample correlations. So it looks like this, right? And that's good. That's kind of what I wanted. I wanted to see here, most of them are correlated, like all my theta cells are correlated with theta cells. I see there's correlation again between my luteinized, right, with my other luteinized samples. They're highly correlated. And this tells you there's something special about granulosa cells, right, and how they behave. And then the other sets, the selected and differentiated, have a tendency to correlate with each other. So something not horribly outlier-like. And again, I can just click and make that a little bigger to see all of my little labels there. And we'll see that there might be some oddballs here, like this one granulosa, luteinized cow. That one individual may be a little off, right? We can kind of see this own little line there. Or maybe another little differentiated cow, maybe a little differently correlated than the others. But that's about it. And nothing like too deviant. If we look at the range, right? 0.85 all the way up to 1 for our correlation, that's pretty reasonable, right? As we think about it, it's reasonable enough that there's nothing to lead me to think that I want to exclude one of these samples. So we'll keep it, right? So that's a nice little thing to keep track of. So that's good. Everything looking is happy right now. I'll leave it alone. Again, just relate to what we've been doing. That's that sample quality control that we've been doing right here, right? We've done any filtering that we've needed to. Now let's move on to kind of modeling on our gene expression, differential gene expression, right? So I'm going to close that transpose down. I don't need to save that. Okay, we'll get to you. Let's go to that modeling. This is pretty straightforward. We're going to use something called the response screening platform. And it's been here for a while. Most people probably didn't realize you could do it, but it's explicitly designed for this purpose, right? We've just enhanced it recently to make it a little bit more amenable to differential gene expression. And so the idea is, what we want to get out is, well, basically we want a volcano plot. And we want our means differences, right? And I'm going to do this by granulosa and by theta. They're really independent tissues. We should be treating them independently, right? And so here we are. We have means differences. And we have the values too. Our false discovery rate corrected p-values. This is the Benjamini-Hochberg multiple test correction method. So here are the p-values for each of our comparisons. And this is all possible combinations, right? All possible combinations here. And we call corrected for that false discovery rate. So that's great. We have differences. We have our p-values. We've got this general volcano plot. That's nice, but this is everything for granulosa. So when we have something like this, it might be a little bit more difficult to tease out. Some of you might be asking, like, how did I get here? This is great. It's under analyze, multivariate. Well, sorry, we'll get there. Screening. So analyze screening is where we want to go. Response screening. And here we are. We have 22,500 responses. Now, I could do this manually. And I've had said people in the community ask, questions like, I want to use fit Y by X. And I've got my one transcript by, my stage or by the stage animal comparison. And I say, okay, great, right? I'm doing ANOVAs here, right? We're converting it into a one-way ANOVA instead of a two-way ANOVA. And that's great. You could do that. But you got to do this 22,000 sometimes. So this is ridiculous. You don't want to do it that way. So that's what this platform does. It does all of that. And so that's all I've done. The only difference here is I've taken tissue and I did this as a by, right? I've done it as a by group like here. There's various other combinations. It might help if I actually put this here. There we go. So I've got my X, just like I did that fit Y by X. You could do it by groupings and it will pair everything in one table, and that's nice. Just to kind of a quick overview of things, that can be useful. I could do each with a control or all combinations. I chose all combinations because there's no explicit control here. If you do with each with a control, It's the first thing that's in the list. An alphabetical list becomes control, but you can actually define that with what we call a column property. And that allows you to define what the control level is, and it'll be everything to that. So that's a convenient way to do, with control. And that's how we got this, right? And we're done. Seconds later, we've just done 22,000 comparisons, 22,000 tests, I should say. And there we have it. But as I said, this is great. But we might want to do something else. This is great. We're done. It's very simple. Now what? Right? So we might want to look at the individual comparisons. We might want to visualize. We want to do some filtering and clustering. Maybe we want Venn diagrams. We can take that a step further. Like, we only care about certain combinations, right? We want to compare to cow. Actually, cow is our control level, right? And heifer is considered the young or different metabolic state, and cow is a different, and cow is going to be our reference. And we only want granulosa only and theca only, differentiated. So we want these specific 6 comparisons only, not all the others, right? So we want to limit ourselves. We also may want to limit based on thresholds. We'll get to that in just a second. But what I may want to do first, first I may want to show all my columns. And notice I don't have all my columns here. Sometimes it's nice to show all of them. If you right click and say column selection, I can expose all of these columns. Why am I doing that? Because we're going to right click and do something called make combined data table. And then it will do that. And here's our table. So we converted those 2 things. Here's our tissue. Here's our level, right? Here's our this versus this. That's what level versus, that's what we call minus level. And we have all our other information, right? So that's a lot that we have there. That's perfect. And the reason why I want to do that, because now I want to filter, right? And if I do this, I made a mistake here. What I'm going to do is I'm going to do this instead. Go to graph builder. I'll manually do it for you. I can put my difference, an X. I could put my false discovery rate. We'll call the FDR log worth. This is the negative log 10 p-value. We want data points. Seems like it looks nice. We're not done yet. I'm going to add something called local data filter. And what we want is tissue. We want these, and we probably want this and the difference. Great. So I want granulosa, right? I want we're done over here. I can then take this and say I want differentiated heifer versus differentiated cow, right? That's all that's going to be exposed here, right? And I can adjust all kinds of ranges too. I can set things up however I want, right? And then you can see it's already colored like red in here as well. I can make little lines, double click on axes and say, well, let's see. This is going to be 1.3. We'll see a line there. So that's kind of a threshold of some sort, or I can change that. Remember, this is log 10 space. So if I wanted 0.01 p-value threshold, I would just say 2. And I would see that, and we could see it makes sense with their colors here. So I'm drawing my own line here, and I could also do it with differences. In this case, it's transformed. I could say something like, it's kind of a log, it's our log space. So we're going to say it's 0.585. Let's add a line. And let's add another one at negative 5.85. Right? So we're going to roughly log 2 of our differences because we're in log space. And now I've got my goalposts, right? So now we can see those that might be interesting here and those that might be interesting here that are underrepresented or overrepresented relative to cow. And then I could continue on down our next pairs. luteinized heifer versus luteinized cow, right? And I can see those that might be significant. So yeah, another way in which I could explore each of these individual tissues and the different comparisons, that I want, even filtering, right? I've added the differences and I've added the filtering here for log worth, right? So that's there. So it's pretty cool. And I've done all this, kind of by hand. I want to keep this off to the side here for a second, right? And that's what my attempt was for this kind of scripted graph, but it broke. That's my fault. We also may want to do some filtering, right? I don't want all 675,000 comparisons. So I only want those that have an FDR p-value of less than 0.05, and those that have a difference that's less than or equal to, negative 0.585 or, 0.585, right? I could do that. And we could do it a couple ways. One is just select where, right? So I can do a lot of these kinds of selections here. So we could do it here. We could do it here as well, where if I had my subset, right? So there's kind of select rows or filtered rows. So if I filtered things, I could subset that way, right? So there's all kinds of filters I could add based on these things. Another way I'm going to bring back your attention. Here we had all this original table. One interesting thing that we can do is use something called a select where clause, right? And we can make these kind of complicated versions instead of manually doing this multiple times. I could do it. And that's kind of what I did here. If I were to copy and paste this is a good example of this. I could come up here and just for my granulosa here, I could say select where, and paste this in, and then it would find them. Unless I did a little difference there. Sorry about that. But I could do that. I can make these complex selections here. And then I could say select columns and then subset, right? So there's a variety of ways I can do that selection. So I've done that. I'm going to move some of these things. Let me minimize this, minimize that. So what happens if I do that? It would help if I don't have this one. There we go. That's my mistake. I've combined this table too many times. Untitled 2. And just close that down for a second. And there we go. There we go. Here's my combined data table. There it is. Now when I subset, right? If I do that every single time, did every single one of those combinations, and I can then just use a simple concatenate to combine them all together, which I've done here. We've got one now. And that's all I did, as I combined all these subsetted things, right? And now I want to get ready for a Venn diagram, right? So I've already done all these filtering. You'll see here that I've even I want to create based on this here, I want to create what we call a kind of a column that's my signature column. I want to designate it with significant by just giving it a 1. And you could just do that. New column, if I wanted to, significant column, and just call it constant 1. So they're all significant. Now, why would I do that? You're going to see here in just a second. What I'm going to do is I'm going to make that column and then I'm going to split it out, right? So I'm going to take all those things and I'm going to make this kind of matrix, those things that are significant. So I took that, right? I took this thing and then I used split, and I split on that significance indicator, right? That's what I did, because you see I've created it. I split on that, and And anything that was missing, I just recoded it to 0. And there's a handy tool that allows me to, if I go to columns called recode, we'll have to select something first. If I say a column standardized attributes, I could recode it. So anything that was missing, I could claim as 0. That's pretty slick. Now these are all zeros. And now I'm ready to make a Venn diagram, right? This isn't added. It's not a built-in tool within JMP right now, or JMP Pro. But if you can download it, you can just search for my community page. It's a Venn diagram add-in. All I need is a single table of 0, 1 variables, which I've done. And now I can say, here are my granulosa ones. I can add them and then say, okay. And now I have my Venn diagram. And then I can do that again, right? Remove them, do that with my theta cells, and then okay, right? So now I can see them, and I have a lot of options and learn more about this particular tool in our community site. But there's a lot of neat little things I can do. More importantly, if I were looking for granulosa differentiated heifer versus cow, I have 60. I can select them, right? And then I could subset these selected rows. Now that I've done that, I now have my gene identifiers, right? And I can see that they're all significant for this comparison. And if I wanted to, I can go back and then find out which pairs, what's its p-values, things like that, because I already had that table a while ago. And that would be this one, right? So I know which ones they are. I have my gene identifier, and now I can join them in, or do what we call a join, and get all those expression values out. And that just allows me now to save out. And just as an example, here's where I get to do what we call join. I like to show a little bit of this. I'm combining it with this list. It's my Y versus my Y. It's my identifiers, right? And then I say, okay. And now I have them all joined in. Here's all my data. I can now export that out. This is a Mac, so I'll use export and save it as an Excel file or a comma delimited file. And now I can use that for additional tools, bioinformatics tools for interpretation, right? So that's a nice little workflow. Now, it took a long time to do this one because this has a lot of more data manipulations that I wanted to show you what's possible. It's kind of common that people will do these things, these kinds of analyses. So I went through that the whole step. One thing I didn't show, let's cancel out of this, is, right, I could also cluster all these things. So I could transpose these things, right? Right, so all I did is transpose these because now I just want to cluster the significant ones and then cluster those, just the significant genes. So I use all that filtering up front, then clustered them. And then I'll see there are sets of genes that are kind of meaningfully different between the two tissues, which is probably tissue specific differences, right? And then within that, you might see some small subsets too. Within those tissues, you might see those things that are probably luteinized. Sure enough, those are genes that are associated with being a stage of luteinized versus the others. And that's just, again, good to know. You can see these kind of obvious differences here too. And that's what I have to show you for this. I'm going to stop here for a second. What questions do we have, or maybe unanswered, Ross?
Hey, Chris. Yeah, I think we have a couple that'd be good for you to comment on briefly. First off, pretty high level one. Are all of the genomics tools in JMP only for gene expression, or can they also be used for population genetic or genomic data?
That's pretty good. So what we'll find out is right now, the ones I've showed you as gene expression. My next use case, which is pretty quick, we'll start talking about genetics. So starting with allele or allelic information, and we'll be able to use things, which you'll see here under genetics. So basic marker statistics, right? Just get your Hardy-Weinberg equilibrium tests. We'll do that. You'll see that we're going to go back to response screening again to do our GWAS. And then I'll mention briefly that we can go to fit model to start doing some more of these more sophisticated. Do I have a random variable? Do I have another fixed effect that I want to look at and get out those those genes, those genotypes that might be most significant, and build even a quick Manhattan plot for that. But we will be continuing on. From a population structure, you can use PCA. We don't have relatedness measures yet, but that is coming. That is coming in the next version, different relatedness measures calculations, which means we're going to QK mixed modeling in the next version.
Great, thanks. And one other one quickly here, just for clarification, can you upload a database, for example, Silva, to compare your samples to similar microbiome analysts?
You can connect to a database, right? So these are all, connect to a database, but not like a public database. So you'd probably want to download that. It would become its own data set. You'd have to have this one enormous data set and then try to do that comparison. But if you're going to do kind of like phylogenetic type analysis, this would not be the right tool for that. So it's not like KIND, for example, which would be a much better tool for those kind of microbiome type analyses. But if you get the OTUs out, then you're going to go back into your statistical analysis, and this would be the right tool for that.
Great, thanks. And I think that's all for right now. So why don't we move on to the next example?
Oh, you got it. I did have a quick question here too. Can you get a copy of this journal and data sets used? Absolutely. When I'm done here, I'm just going to take the original data set and the individual journals for both of these use cases, and I can provide them for you. We also have a nice website, which I'll point to just at the end here, that there's some more examples to follow along with as well, that also has links to data sets. So I'll make sure I'll point that out. All right. Okay. No, not yet. I get the one little question mark real quick. Will we be able to check for population admixture? Not yet. But that's a great thing to put on our wish list, which I'll point to as well. If there's something you don't see, let us know. We have a wish list. We pay a lot of careful attention to what people do. And so that's good. So let me kind of clean up here. And so that's differential expression. And let's close some of these things up. I think it's just about that. All right. Close all. There we go. So that's that one. And I think there's a couple other questions we'll get to in just a second. So let's do a genome-wide association tests. All right. Oh, and here's a gene expression one I want to make sure I answer. So co-expression network analysis, we don't do co-expression network analysis yet. Again, you could do correlation between genes, but not from a network point of view. All right. We'll get to example. I'm going to leave that. There's a good question there about samples, so we're going to get to that one. So let's talk about genome-wide association tests. Again, just a very simple example. This workflow, it always seems more complicated. When I can block this out, it always seems really complicated, right? We have a bioinformatic process of aligning to a reference genome, calling out alleles, right? JMP Pro prefers numerically coded genotypes, but you're not limited to that, which I'll show you. But then we might need to recode them, because like I said, we like to use numerically coded allelic genotypes in allelic format. But we might want to do some basic things, or marker statistics. We want to maybe look at some bar charts. We might want to look at linkage disequilibrium or linkage decay between different SNPs. We might want to know about the Hardy-Weinberg equilibrium tests. If they're out of equilibrium or not, we might want to filter those out. We might want to recode major and minor allele designations. We might want to look at heterozygosity, minor allele frequency, missing proportions, right? We might not do a lot of that. So we might have to recode to major and minor if necessary. We might want to be filtering, right? So we do all of this upfront work. That's kind of our kind of QC steps here. And then we want to do our modeling, a SNP trait association test. And again, you get some Manhattan plot. We might want to look at some distributions. If you want to look at volcano plots, you possibly could, right? And then you filter and then save out, right? So that workflow looks complicated, but it's more of a mental workflow. and it's not as many steps. So we're going to take a look at Bernese mountain dog sarcoma as an example. And this is very simple. It's a case control, right? We do have kind of a covariate, if you want to think of it that way, as a region. So there's the European dogs and there's the North American dogs. We've got quite a number of samples, right? So 128, case, 114 case for North America, right? So that's about 242 total case. And for total control, we have about 232. So we got a large number of things to look at, and we really, the article set it up as kind of, let's look at these things independently, and then let's look at them combined. So we can kind of do that if you like. We'll kind of look at those that are independent at first. Some things that we'll need. This one's not too bad. I just kind of got the annotation downloaded. This came from Geo's website. So I did that. And then the use cases themselves. This actually came from the you look at the Geo series matrix files. We can actually import those. This is kind of what happens when we do that. So I've got this. Let me keep my annotation off to the side here for a second. When we import a series matrix files, it's just kind of file, open. There's some more descriptions here. We get a bunch of stuff. And we've corrected this in the next dot release to automatically bring in the A's and BBs here to do that. We've got all of our nice information here. We have our phenotype, which is really our case can control, our breed, whether it's North American or subregions in Europe, right? And then we have our genotypes here. So here's our samples. Here's our SNPs, these not so cleverly labeled as 1 through 22,362. That's their IDs, very generic, not meaningful, but they're there. And that's what we have. And I need to get this into here, right? But I may want to recode this and transpose it first. So this is simple enough. What I'm going to do is I'm going to transpose this. This is just how I get it from Geo. Your data set might already be transposed, which I've seen most often, but mine isn't. So I'm going to do that, right? This is simple enough. Here's our label, right? There's even a little auto refresh, which is nice to see that real quick. And this is kind of a Geo accession. Great. Done. It's already mapped to it. I don't need that, right? And then I'm going to do something here. I'm going to recode all these things. And this is kind of fun that I can go in and say standardized attributes. And this is where that recode is kind of nice. I can recode all of these things as, well, let's just call them for right now 0. And we're going to call that one 1. We'll call that one 2. And we'll call that missing, right? We've got that. Okay, we recode. They're all done. Oh, and you know what? Let's make them numeric, right? They aren't numeric, but they're considered like character at the moment. So let's standardize that to numeric, and we'll just call it continuous for the moment. Done. That's it. We've now recoded these things, and I haven't paid much attention to what's major and minor at the moment. If it was AA, I just assumed it was major homozygous. If it was 2, it was minor homozygous. And if it was AB, that it was heterozygous, so we gave it a 1, right? So that's what I did for now. Then I'm just going to update this. So we'll use our handy update tool, and we'll update this. Here's our, remember our geo accession with our geo accession, and that's it. And we're done. We've just updated this. So now I have my functional data set. I don't need that anymore. Good. So that's what we're working with, right? So we have 22,000 SNPs. We've done a little data prep right there. I've already stepped through this. I made little scripts for you, merged them. I want to make some additional things. I want to recode case control, right? And in this case, I've done that. And just as a tip, you can always go right click on a column and say recode. And I just call this condition, just to let you know how I did that. Right? And I would just call this case, control, or 0 and 1, or 1 and 0, however you want to call it. And that's all I did. Right? I want to discard them. And then what I did is I also recorded North America. I call this thing called region. So I took the origin. If it was North America, I kept it. If it was not North America, so someplace in Europe, I just called it Europe. That's it. Now the designations, right? And then color and mark by condition and region. That's all I did there. Again, that was easy enough. It was color and mark by, region, right? And then I came back and then I marked by condition. And so that's okay. I just multi-step process, did the same thing. So we're ready. The QC part is easy. It's marker statistics. We have it here. Oops, I did something not right. I had my annotation off to the side here. Here we go. So our marker statistics, let's do it manual. I'll show it to you. Here, that's not what I wanted. Here we go. Marker statistics right here. We have all our genes. There are markers, right? We can use an annotation table. We have our ploidy. This is this is diploid, but you can have multiples. You could do some, if you did, marker by marker, that's where you get the linkage decay or linkage disequilibrium information. We're not going to do that here. I'm just going to say, okay, what we'll get, I'm going to choose an annotation file, which I've already opened. So my chromosome position is my annotation group. My position is annotation position. And our ID ref is the ID of the columns. And now I'll have this, and I've done something really crazy here. What did I do wrong? This is how I know something's not right. All right, let's take a look at this real quick. No, well, it looks like, oh yeah, I did this right. I did this intentionally. I did this for the whole population. I didn't segment them in different regions, right? I didn't do it as North America and others. So I did that first. So I've got all of that. And here's what I want to do. Most commonly, what I wanted to point out is most commonly coded as 0. If it was not, we would see things like this, right? For example, and this is what tells me I have done something horribly wrong here. Because my annotation is off somehow. All right, so let me back up. Let's see where I went wrong. That's why. I should have, everybody should be there. Oh, I'm missing a few things, aren't I? That's kind of a problem. Let's try that again. It's always a good check to see what happened. Apologize for that. We'll put this back into its happy state here. We'll recode them properly. I think this was my mistake. I didn't get everybody. Try that again. It'll take a little bit to recode things. Let's make sure I've done everything. Okay, good. Again, we want them numeric and continuous. Yeah, I'd only selected a few. That's my mistake. I apologize for that. Always double check. Everything looks okay. Let's go back to our update. Ooh, There we go. Verify real quick. All right, good. Everybody's back where it should be. All right. I'll do one last thing I forgot. Apologize for that. Wanted this, and there we go. So we go back to our marker statistics, and this is what it should have been, minus my mistake. So sorry about that, but now I have everything I want. So I have our minor allele frequency, right? We have our minor allele. If this common allele is coded as 0, that means the most frequent allele, is incorrectly coded as the most common allele coded as 0, right? Is not. B is actually coded as the minor allele, as the major allele. We need to recode that. So simple things, select where, right, equals 0. Right, and these are all there. I can now select my columns, and now I can recode. Now, those that are actually by frequency in the population that we're looking at, right, which is really, it's a subsample of the true population. The most frequent allele will now be coded as 0, and the least frequent allele, being B here, would be coded as 2. So now we have that as well. We also have our Hardy-Weinberg equilibrium test, right? So we have those p-values, and there. And so this is like a Manhattan-like plot, but we may want to take that out. We might want to keep that, and we want to save what we call the results table, which is just this. It's just the p-value. We say results table, and then I just renamed it as marker statistics table. And the nice thing about this, so we got this marker group, right? Problem is I need to recode that because it thinks -1 is really the X chromosome. And it'll say, yes, I'm going to turn it into character. Let's do that in place. Recode, great. Now, why did I do this? Well, now I can make a Manhattan plot. That's why. That's easy enough. Here's our FDR of the Hardy-Weinberg equilibrium test, right? I can then say, here's my group, and then here's my position. And we're not doing points here. We want our points there. There we go. And we can easily color them by our group. And there's our basic, our Hardy-Weinberg equilibrium, Manhattan plot. And if I wanted to, I could separate that out by region. So simple enough plot. Basic QC, there's a lot of things how to equilibrium, so we're going to probably want to filter those things, probably due to some inbreeding. So we'll do that. We just filter them, right? That's simple enough. And what we do when we filter them, right, it's not hard. We just kind of say, here's our main table. We go back into here and just say in our main marker statistics and just say select where, right? We can do it by minor allele frequency. We can also do it and by, this FDR log worth. Select them, right? Select the columns, and then I can say exclude them, right? I just kind of group them together and say exclude. That's all I did, right? So I did this little and principle here. And so that's it. The rest of the GWAS is just response screening, right? And just like we did before, right? Response screening, the difference here is they're X's and our condition is Y. That's the only difference we're doing here, right? And then we screen through all of those. But that's pretty much all we're doing to get this result. In this case, since I had numeric values, we're basically saying these are logistic regression. So I can click here and say fit selected items. And we'll see now that as we go from the major to minor, right, there'll be more cases than there are controls when we get to a minor allele frequency. I mean, a minor allele frequency, the minor allele. And the opposite, right? We have more case than control for major allele to minor allele for that particular SNP, right? So this is one way to do it. That's if you keep the SNPs as what we call numeric. But if you convert them into kind of categorical, then you're going to get a chi-squared test. So the latter is the allele test, right? The former is the trend test. It's just a matter of how you designate these as categories. Then again, after that, it's just a matter of save the p-values table and update with annotation, right? That's all I've done here. Just kind of join those two things together. And then our Manhattan plot. And now, since we've filtered out all those things and we've done our case control, association, we'll quickly see that in North America, that's chromosome 11 has a lot of associated SNPs with case and control. And there seems to be some more European specific ones in 2 different chromosomes. And since we selected these, right, and again, it's up to you, just like the gene expression. We can save these things as a subsetted table and export them out and do all of that as well. And that pretty much was it as far as what's possible at a basic level. One thing I do want to point out, I did this all using response screening. You could have used something called fit model. And if you do that, there is something called, you'll see it response screening at the bottom. And you would still have your kind of condition. You'll notice it says, oh, I don't like that. I'm going to switch it to nominal logistic. That's okay, because in this release of 17.1 that you have, we have something called switch, which is nice because now you can take all of these and it effectively does the same thing as we did in response screening. So each SNP it loops through each one of these. And the nice thing about doing it here in a nominal logistic, I can include my region as another main effect, right? So I've got another fixed effect. So here's my region per SNP, right? And then I can do that comparison. And my target level is control, which I think I like. So now if I run that, right, it'll go through. Oop, and I forgot to do something. That's my fault. I think these options, there's some options on the red triangle that says, give me the p-value table only. So I think that's awesome. You'll see that you can use mixed models in here as well. So you can define a random variable. You could do PCA and put them in here, but it's not quite QK mixed modeling yet, but we'll get there. All right. And so that's what I wanted to show you. I'm sorry I'm over a few minutes. It's my fault for making a mistake initially. I would have kept us on time if it wasn't for that. But I do want to show you one thing before we go. And that is what I want to show you here. Oop, I didn't show this in the window. Additional resources. People would ask, are there additional examples? There is a data set. You could go under sample index, but go here. There's this thing called genomic analysis with JMP Pro. It's nice in that it steps through these. All these platforms were talked about. It's got 2 additional case studies that you can look at. Talks more about predictive modeling, more pattern and structural discovery, things that I haven't covered, just more data sets to look at, different types of things to import. So that's great. Be on the lookout for an upcoming version that we are going to have. We are going to be able to do QK mixed modeling because we'll be able to do like identity by sharing and some couple of other methods. We will be able to do QK mixed modeling to do things. For those people who are doing G by E type stuff, you can do that now. That's basically it's basically an ANOVA. You just use a mixed model. Define those things are either random variables to get your BLUPs, or your parameter estimates are going to be your blues. If that's what you're interested in. So that's possible too. The only other thing, I think I can show this. Yeah, I won't do this. There are one other things I want to point out. I won't be able to show you, but under multivariate methods, we do have something called multivariate embedding. So for people who are looking for a higher kind of structure, we do have TSNE and UMAP, directly implemented, right, natively implemented in JMP. So you can get either 2 or 3-dimensional views of these things. Go ahead and take a look. I think we might have a blog out for that. I'm not quite sure, but that tool is in there as well.